Hello all, welcome to the HL Bio lesson. This relates to carbohydrates and lipids today. We'll be mainly focusing on the structure of carbohydrates and lipids, and we'll be leaving sort of the ideas related to energy and health till next lesson. Our essential understanding for today is compounds of carbon, hydrogen, and oxygen are used to supply and store energy. So both carbohydrates and lipids have only carbon, hydrogen, and oxygen. Let's look at carbohydrates first, and here we're, as I said, mainly focusing on the type and the structure. Our first understanding is this. Monosaccharide monomers are linked together by condensation reactions to form disaccharides and polysaccharide polymers. So we can see that key idea of condensation reaction again, as well as those key phrases of monomers and polymers. The monomers of carbohydrates are monosaccharides, a single carbon ring in that structure. And these monosaccharides can be classified by how many carbons they actually contain in that structure. So for example, trioses have 3 carbons with this formula. Pentoses have 5 carbons with this formula. And hexoses have 6 carbons with this formula here. So glucose would be an example of a hexose. Now, as you can see, there's a bit of a pattern to how this formula is devised, with the number of carbon and oxygen being the same and the hydrogen being double that number. Another key word moving on is isomer. Now, isomer means that different molecules have the same chemical formula, and we'll see that this is important for both carbohydrates and fats moving forward. For example, an isomer of an isomer would be the hexose monosaccharides, glucose and fructose. Now these are isomers because they are different structures, but they both contain the formula C6H12O6. And here we have them up here, glucose and fructose side by side. Okay, we won't spend too long on the actual condensation reactions because I feel this would be covering territory we've already looked at, but here is an example from the blue. Glucose plus glucose joining together to form maltose, plus water from that reaction. Now importantly, the bond that's formed between these two monosaccharides is called a glycosidic bond. I'll put that formally into the presentation in a second. Condensation reaction showing the formation of the disaccharide maltose. One monosaccharide donates a hydroxide ion, the OH group, and the other monosaccharide donates a hydrogen ion, the H plus group, which then form the water molecule. The free bond then forms a covalent bond, the sharing of electrons between two monosaccharides. So we can see here the OH group and also the H group, and these two combine, electrons are shared, and we result in this bond, this glycosidic bond, through the oxygen. We can also see here that the bonding is between elements on carbon number 1 and carbon number 4. So you can see here these numbers refer to the carbon number in this molecule, starting from the right hand side and moving to the left. 1, 2, 3, 4, 5, 6. So the bonding between these two monosaccharides is through position 1 and 4, so we call it a 1 to 4 linkage. And that's shown just here. Okay, moving on, let's just quickly have a look at some common disaccharides. So first of all, we have maltose, which is malt sugar, often used in brewing. And this is formed from 2 glucose molecules joined by an alpha, so alpha glucose, 1 to 4 glycosidic bond. So we can see up here this characteristic bond formed between position 1 and 4. In sucrose, which is essentially table sugar or a transport sugar in plants, it's formed from two different monosaccharides, glucose and fructose, bonded between the alpha 1 to 4 glycosidic bond once more. And we can see here the third one, the third disaccharide we should focus on is lactose, which is the sugar found in milk. It is formed from galactose and glucose, again joined at the 1 and 4 position, but you'll see the bond is slightly different because we're dealing with beta glucose in this case. This table is quite good. It summarizes the functions of major polysaccharides. We'll look at the structures and you'll learn more about the structures in class. But essentially, polysaccharides are long chains of repeating glucose units joined by condensation, and they produce a wide variety of polymers. And these polymers of carbohydrates are called polysaccharides. So the three that you need to know for the IB are cellulose, which is the major component of cell walls, starch, which is the energy storage in plants, and also glycogen, which is the energy storage in animals. So please take a moment to note down the functions of these major polysaccharides. Okay, so here is our repeating unit, glucose. I just thought I'd put down a different sort of diagram there. And please note again the understanding up the top, structure and function. Both structure and function of cellulose and starch in plants and glycogen in animals. Let's have a look at some general structural features of cellulose, starch, and glycogen. So first of all, they are all polysaccharides of the same monomer unit, which is glucose, simply repeating that glucose molecule over and over again. The glucose is linked by those 1 to 4 linkages, so from carbon number 1 of one molecule to carbon number 4 of another. Most of these polysaccharides are branched, which means they have a central chain with these side chains coming off, and we'll look at that in a little more detail soon. And starch also has two subcomponents. One is called amylopectin and the other is called amylose. Amylose is the only one which doesn't have any side branching. It's in a linear chain, which is quite interesting. All of the rest are branched chains. And each polysaccharide, hence the name poly, is made up of thousands of glucose molecules. So the term polysaccharide there means many sugar units, and they're glucose in these examples. Nice simple little diagram here. We can see the structure of starch. So we see amylopectin there, which is a branched molecule, and there's our linear amylose chains. Now we'll look more at how those sort of interact to form starch in class. Glycogen, you'll see, is very similar to amylopectin. This is the storage for energy in animals. Again, lots of branched chains. And we can see cellulose is very, very different once more. Even though it's composed of glucose molecules, a very, very different structure. So part of our classroom application will be looking in more detail at these structures. Okay, let's move on to lipids. And again, we're focusing more on the types and structures, not really dealing today with the energy or the health implications. We'll deal with that in class. So first of all is the understanding that fatty acids can be saturated, monounsaturated, and polyunsaturated. So probably a little bit of review here for our Bio 2 work. All fatty acids have a carboxyl group at one end. So this is the characteristic end which has a carbon bonded to an oxygen and also bonded to an OH group, an alcohol. And at the other end we have a methyl group, which is our carbon bonded to 3 different hydrogen atoms. In between these two ends, the carboxyl and the methyl, we have a long chain of simply carbon and hydrogen. Now this is between 12 and 14 carbons long. So here is our picture from the book, this one. It's nice and simple. So we can see at the left-hand side, we have the carboxyl group, which is our carbon bonded to an oxygen and also an OH group. In the middle, we have our hydrogen-carbon chain. And at the end, we have our methyl group with the carbon bonded to 3 other hydrogens. You'll see the label omega N then. When we start to talk about the different types of fatty acids, this becomes important. So just remember the methyl group is the omega end. Let's have a look at saturated fatty acids. Saturated, just like something is saturated with water, for example, it means the carbons are carrying as many hydrogens as they can. They are saturated. There is no more positions for hydrogen to bond to. This essentially means there are no double bonds between the carbons because every single bond is taken up by a hydrogen. It forms a straight chain with no bends or kinks. And it's usually found in animal products, for example in butter or the fat in red meat. And these are generally solid at room temperature. So this is the diagram we saw before. We have our carboxyl, our methyl, and our chain in the middle. And butter or the fat in red meats is a good source of this, and I'm getting hungry just watching this. That steak looks delicious. Okay, moving on. Let's have a look at monounsaturated fats. Now these include one double bond, and it creates a bend in the molecule. It's called unsaturated because it actually has 2 empty spaces formed by this double bond where hydrogens in theory could attach to. So this is why we call it unsaturated fat. Here is an example here. So you can see the double bond in the pink box, and you can also see this characteristic bending of the molecule. Polyunsaturated fats, as you can imagine, the term poly means that it includes more than one double bond. It's found in plants. A good source would be, for example, olive oil. And they are liquid at room temperature. It has many bends, so if you have lots and lots of these double bonds between carbons, you tend to get this characteristic curving or bending shape. You can still see there the methyl group and the carboxyl group and this long chain of molecules in between. And as you can see, there's olive oil is the good source of this. So we'll be looking more at the health implications of these in the class. Okay, let's look a little deeper at unsaturated fatty acids now. We'll see that they can either be what's called cis or trans isomers. Now remember, the isomer, same formula but different structures. This occurs during food processing, so where you're artificially manipulating that molecule. And what people do is they remove the double bonds from the unsaturated fats and they add hydrogen to where those free bonds that are created. And this is called hydrogenation. So you might hear the term in media, hydrogenated fats. This straightens that naturally bent shape that we just saw. The natural or curved versions of the fatty acids are called cis fatty acids. So those are those ones with the double bonds. And the artificially straightened versions are called trans fatty acids. Now you might have heard that in the media, negative health consequences associated with trans fatty acids. We'll look at that more in detail. Something else to note is these fats, which you might have heard good things about, called omega-3 fats. Now remember we talked about the omega end of the molecule previously. This is where this comes in. So this is where the first carbon double bond to be found in this molecule occurs at the 3rd carbon atom, counting backwards from the omega end. Now that's a little bit complicated. I'll show you on the diagram in a second. And fish are an excellent source of this. So we can see here is our fish as a source, a nice bit of tilapia. Here is our omega end, and we count 1, 2, 3 carbons back from that omega end, 1, 2, 3, and we get our first double bond. So this is why it's called an omega-3 fat. We tend to focus on these because they have good health benefits. You'll also see there's other types, for example, omega-6 and so forth. But omega-3 is the one they want you to know for IB. I've also included this table from the book, which I think is quite good. We've talked about a lot of different kinds of fats, but here's a nice summary table where you can see the curved or the straight ones. You can see our omega-3 fats, our cis, our trans, our saturated, our polyunsaturated. It's a nice table summarizing everything. So please take time to look at that when you have the chance. Okay, we've looked at this previously, so we won't spend too much time on this, but triglycerides are formed from the condensation of 3 fatty acids and 1 glycerol. So there's actually 4 reactants in this reaction. And of course, there are 3 water molecules produced as a result. I inserted that bit in, but please keep that in mind. So we can see here our glycerol molecule, and we can see our 1, 2, 3 fatty acids, resulting in the triglyceride here, the triglyceride lipid molecule that you can see on the right hand side. And please don't forget about our 3 waters that are produced one each time a fatty acid is joined. Now, that is pretty much it for today. So as I said, we've mainly focused on structure and the different types of carbohydrates and lipids. We have much more to talk about in class, particularly relating to health implications and BMI and things like this. But I look forward to talking to you then. Happy studies.